Good evening, everyone. My name is Michaela Smith Hendricks, and I will be your host for After Meeting the Microbiologist. This is a YouTube video that follows the podcast Meet the Microbiologist that is hosted by Julie Wolfe with the American Society of Microbiology. And this week, the interviewee was Dr. Gautam Dantas, who is a professor at the University of Washington School of Medicine. And the topic for this week was antimicrobial-eating microorganisms and resistomes. So let's talk a little bit about how Dr. Dantas got to an interest in resistomes and antimicrobial eating microorganisms. So Dr. Dantas actually started off studying soil microbes in a postdoc program. He was actually interested in strengthening a community of lignin degrading microbes, but that study actually led to some other discoveries that I'll discuss a little bit later that led to his further career choices. And so he has a variety of interests in the field of microbiology, but most notably he mentions microbial ecology and how viewing microbiology through that lens really lends itself to understanding the environment and understanding how microbes function within different ecosystems. So he feels that that approach allows you to get a broader view of the whole story instead of piece by piece. And I actually agree with him. I think looking at things from a population standpoint, if you will, I'm a population health, public health advocate. So I agree completely that you're only able to understand specific stories after you've looked at the bulk of the problem and how it got to where it ended up. So definitely agree with him on a population whole story approach and not just trying to understand microbe by microbe, but instead trying to understand the dynamics that those microbes play in a population of microbes. But Dr. Dantas' overall interest is microfunctionality and the roles that different microbes play in communities and populations, or microbiomes if you will, and how they evolve and how they continue to live amongst each other. And so we'll understand that a little bit more as we discuss the bulk of the conversation between Julie and Dr. Dantas. In Dr. Dantas' lab, they actually utilize genetic sequencing, which allows them to manipulate the genetic sequencing of microbes that they're interested in, and being able to use that technique, they're able to formulate hypotheses for how they feel a microbe should function within a population or the potential of that microbe. And so the only downfall Dr. Dantas mentioned of specializing in genetic sequencing is that they're not necessarily able to test all the hypotheses they're able to formulate. So meaning they're able to generate a lot more hypotheses than they're able to test. And so he's thankful for us being able to share information so widely across the world with different people, because his hope is that his colleagues within the field of microbiology will take on some of these hypotheses he and his team have created and test them for the validity of those hypotheses. And so that's just a little background about his lab and what techniques of microbiology they use. So now let's talk about antibiotic resistance. So I think that it's safe to say that even a layperson, so someone who's not involved in the field of microbiology or science or medicine, has heard something about antibiotic resistance and how it is a concern for our population, or how it's something to just look into in order to understand your own health and the health of your family members. But the approach that Dr. Dantas and his team took on antibiotic resistance is understanding resistomes. And so understanding that resistomes aren't necessarily a clinical issue, but can be observed in different ecosystems across the board, such as soil. So the idea of a resistome was proposed by Gerry Wright, Dr. Gerry Wright. He proposed this concept of a resistome, and Dr. Dantas actually referenced him in their conversation in his conversation with Julie. And I'm going to paraphrase the paraphrase definition that Dr. Dantas gave during that conversation. So essentially what a resistome can be defined as is a collection of genetic determinants in any random population of microbes, or a microbiomes if you will, that would enable a phenotypic resistance. So essentially what this means is if a gene for genetic resistance is observed within a population or a community, that would be defined as a resistome. So when we talk about the concept of resistance or resistomes, there are 3 hypotheses that Dr. Dantas narrowed down as what are the typical hypotheses that a lot of researchers look into. So the first one, of course, being independent evolution. So that means the pathogen has developed a resistance to the antimicrobial organism. And so that is known as the common one that people look into. So the pathogen now being resistant to the antibiotic. The second one being that resistance between microbes, it has gone back farther than what we know. So meaning that resistance gene within those microbes has always been there, and it was kind of fate for that resistance relationship between those microbes to occur, no matter what we did or how we functioned in society, or how those microbes interacted in their ecosystems, I should say. The last hypothesis is known as the producer hypothesis, and this was proposed by Julian Davies. And so what this hypothesis actually entails is that the same microorganism that is producing the antimicrobials must also be the source of resistance. So this is actually the one that Dr. Dantas and his team felt was most valid, and a great explanation behind resistance and resistomes. And so the explanation that Davies was able to offer behind the producer hypotheses is that the reason that this is taking place is because those antibiotic compounds, or those microorganisms within those antibiotic compounds, are creating, are not creating, but have genetic resistance as a safeguard against what the compound is doing to the microbe. So he explained this further by saying that if there was no resistance present in those antibiotic compounds, then essentially it would kill itself. And so that's the evidence he provides for the producer hypotheses, and that goes into Dr. Dantas' idea of resistance and resistomes being something that naturally occurs within microbes, and can be viewed within different ecosystems such as soil microbes. So I just want to highlight Dr. Dantas' initial study that led him to antibiotic resistance once again, which was the soil microbe lignin degrading soil microbe experiment that led him to find that there were actually microorganisms who were capable of metabolizing antibiotic compounds and using the antibiotic compound to grow and flourish within the soil microbiome. And so this brings us to the most recent study that Dr. Dantas and his colleagues have put out regarding this topic of resistomes. He did a cross-sectional study and collected soil from 3 different states within the United States, those states being Massachusetts, Pennsylvania, and Minnesota. So he chose soil from sites that were not as heavily populated, because he wanted the soil samples to be rare and not have so many different variables introduced into the soil that could skew the results or create different findings based off of other factors. So the findings. Dr. Dantas actually discovered that there was 10 times as many genetic resistant genes observed in the soil microbes than in pathogens. And so the reason Dr. Dantas gave for there being so many different microbes that display this genetic resistance is because soil microbiomes have been around far longer than people, and so he feels that it's just a case of time allowing these microbes to express these genetic resistance. And so still what he found was that soil microbiomes and clinical setting, so human beings, if you will, have similar ecosystems in how they process these resistomes. So the findings of this study was so important because it did show that resistomes and how they function within the clinical setting is very similar with how they function in the soil setting. And finding so many more microbes that had displayed this genetic resistance gene indicates that it may be able to inform some of the functionality of microbes and how they will perform in the clinical setting as time goes on. And so it's just something to keep an eye on and something to think about. And Dr. Dantas has actually already started looking into understanding the directionality of these microbes. So another finding that Dr. Dantas found during his research, during this research project, was that the greatest number of gene-resistant DNA was actually found in the soil in dairy farms where cows had been treated with antibiotics. Because the study was not a longitudinal study and was a cross-sectional study, and because the findings were not statistically significant, he wasn't able to draw that conclusion. But he has already started research on a longitudinal study that will look further into this finding so that he can determine directionality, because he did observe mobilization in the resistant genes within the microbes that he observed. So he knows that mobilization is there, but he just doesn't know what the directionality means. And directionality meaning, in the case of the dairy farm and the cow, he doesn't know if it's the soil microbes that are affecting the cow or the cow that is affecting the soil microbes. And so hopefully with this longitudinal study, he'll be able to find that conclusively, what the direction is of that. And so ultimately what this will do is help us understand how microbes mobilize between different ecosystems and different communities, and will enable us to understand better how we can engineer microbes in the future, or if there are certain genetic resistance or characteristics that we're not aware of that we should be when trying to alter these microbes for our benefit or for other reasons. And so I think it's very interesting, and I'm excited to see where this research goes forward. I'm really looking forward to the longitudinal study that Dr. Dantas and his team does. I will actually be looking out for those results because I'm just generally interested. But in the meantime, if you do want to check out the discussion between Julie and Dr. Dantas, please feel free to visit the American Society of Microbiology and the Meet the Microbiologist podcast. I have left the link in the description box, so definitely check that out. Definitely correspond with either Dr. Dantas or Julie if you have further questions that I'm unable to answer. If you have any questions for me, or you just want to discuss this topic further, I would love to hear your thoughts in the comment section, so I'll look out for those. But in the meantime, that's all I have for you tonight. I hope you enjoyed this as much as I enjoyed talking about this topic, and I will see you guys in the next one. Bye.